Greetings everyone! In this video we will build a cancer prediction system using Python and machine learning. We will use the Breast Cancer Wisconsin Dataset to classify tumors as malignant or benign and explore the complete machine learning workflow from data preparation to prediction. Without further ado, let's get started.
Firstly, let's upload our libraries. And now let's load the built-in dataset and convert it into a readable table for the analysis. Now let's print our data. And here we have it. So as you can see, we wrote load breast cancer to load our dataset from Scikit-learn. And then Pandas data frame is used to structure the data in rows and columns. And then feature names used to column labels. And then target values 0s and 1 are mapped to malignant or benign. And then head to show the first 5 rows in our dataset. And now as you can see, each row is equal to one patient or sample, and each column a measured feature of the tumor. So for example, features are grouped into mean values or worst values. The mean values are average characteristics of the tumor, and the worst values most extreme measurements observed. The values represent physical properties like size, radius, area, parameter, or shape, smoothness or symmetry, texture and irregularity, compactness and concavity. And the last column, diagnosis, is target label, malignant or benign. And as you can see, we have 5 rows and 31 columns. The 31 columns, 30 features plus 1 label. Now let's check for the null values. We checked if any data is missing, but every column has 0 missing values. So as you can see, we have checked if any data is missing in the dataset. Every column has 0 missing values, and we have used describe as this gives us an overview of the all numbers in the dataset. So as you can see, for the count, it gives us 569 for all columns, meaning that we have about 569 patients. And the mean radius equal 14. These most tumors have a radius around 14. Standard deviation, this shows how much values vary. For example, small standard deviation values are similar, or large standard deviations values are very different. And for min and max, this is the smallest and largest values. So for example, mean area goes from 143 to 2501. Some tumors are very small and others are very large. And then here, 25, 50, 75, 25, this is low values, 50 median, middle value, and 75 higher values. This helps us understand how data is spread. Let's run our code. And here we have it. So as you can see, this is a correlational heat map. It's a way to understand how different tumor measurements in our dataset. It shows us if increasing one measurement, like radius for example, typically means another, like area, will also increase. This deep red represents a strong positive correlation. If two features are red, it means they are highly related. When one increases, the other always increases. We have also the deep blue, represents a strong negative correlation. So, for example, as one feature increases, the other tends to decrease. So, here for example, the diagonal red, this is solid dark red line running from the top left to the bottom. And this is because every feature is perfectly correlated with itself. As you can see, here is texture error. And if we go down, texture error too, and so on. And now, let's perform the diagnosis distribution. So, and here we have it. As you can see, we have the y-axis for the counts, x-axis is the diagnosis, we have malignant counts and benign counts. And now, let's make some data pre-processing. Let's select features and target. So as you can see here, we have splitted our data into input and target. X for all features, tumor measurements, and Y about our prediction. And for y equal y.map malignant and benign, here machine learning works with numbers, not text. So we convert malignant into 0 and benign equal 1. And then so as you can see, here we train our test split, test size 0.2, which is 20%. So 80% of the data would be training model. We learn from here, and 20% is our evaluation here. So this checks if the model can work on unseen data. And now let's make random forest model, which is a machine learning algorithm using many decision trees. We combine multiple trees to make better predictions. And here we have it. And now, let's make our predictions. Select X test. This guesses diagnosis for the test data. Now let's evaluate our model. One hundred for 100%. 0.2f. And we put the percentage. Let's run our code. As you can see, our model accuracy is 96.49%. Now let's make our confusion matrix. And here we have it. So, here, confusion matrix, y test, y predict. This creates a table comparing actual values, which are the true answers, and predicted values or model guesses. Heat map to make the heat map. Annotation or annote true shows actual numbers inside the boxes. FMT display numbers as integers. X-axis predicted values and y-axis actual values. We have top left, top right, bottom left, bottom right. The top left here, this is correct benign predictions. Top right, benign predicted as malignant error. So the model here says it's malignant although it's benign. Bottom left, here, malignant but predicted as benign. So the model here says this is benign although it's malignant. And it's 1. And here, bottom right, correct malignant predictions. So it's malignant and the model predicted as malignant. So for example, here we have 40. These are true negatives. The model correctly predicted malignant 0 when the tumor was actually malignant. And here 70. These are true positives. The model correctly predicted benign when the tumor was actually benign. So together, these both are 110 cases. These are the ones the model got right. And here 3, false positive, type 1 error. The model incorrectly predicted the benign when it was actually malignant. And here, bottom left 1, these are the false negatives, type 2 error. The model incorrectly predicted malignant when it was actually benign. And now, let's print our classification report. And here we have it. So we have precision, recall, F1 score, and support for 0 and 1. And here we have the accuracy, macro average, and weighted average. For the precision, it shows how many predicted positives are actually corrected. Recall, how many real positives the model correctly found. F1 score, balance between precision and recall. Support, for the number of samples in each class. So for each class, malignant, benign, it tells us how often the model is correct when it predicts that class precision, or how many of that class it successfully finds, which is recall. A combined score of both is F1 score. So precision 0.98, when the model says it's malignant, it's correct 98% of the time. Recall is 0.93. It correctly finds 93% of the all real malignant cases. F1 score 0.95. Overall strong performance for malignant detection. And support 43. There are 43 malignant cases in the test data. And now, let's test the model with new data. Let's run our code. As you can see, the model says the tumor is likely malignant. But it didn't say tumor, it said tumor. What's tumor? No one told me anything. Why no one told me to correct it? Okay, the tumor is likely malignant. Now let's visualize feature importance. And here we have it. As you can see, feature importance plot. Firstly, Look at the top of the chart. Features with longest bars, like worst area here for example, and worst concave points, are the model's most trusted indicators. In medical terms, this suggested that the maximum physical size and the most irregular shapes of a tumor are the strongest signals for identifying malignancy. Second, notice the color gradient. The color serves as a visual guide, where the darker purple indicates high impact features, and the lighter green or yellow indicates features that the model barely uses.
And here we've reached the end of our analysis. We've successfully built a cancer prediction system using Python and machine learning. I will leave the link in the description of the video that helped us to do this analysis. Thanks for watching. See you next time.